Hi, this is Amanda Kocher, and I am going to be talking about the action potential of cardiac contractile cells today. So I want to start off with a little bit of review because typically students would have nervous system and the muscle system before they would encounter the cardiovascular system, and so I think starting with what we know is a great place to start. So when we talk about action potential in skeletal muscle tissue, or when we're talking about nerve impulses throughout the nervous system, we look at action potential and typically follow something along this general outline for a graph. Our typical resting membrane potential around -70 millivolts. You have some slow depolarization that happens to get to that threshold, and then you have rapid depolarization that peaks out at about +30 millivolts, which then inactivates those sodium gates that open, and your cell is able to repolarize as potassium voltage gated channels open and allow potassium to come back into the cell. So all of that happens very quickly. You're looking at a time frame of 2 to 3 milliseconds from start to finish, very quick, and requires multiple neural stimulations in order to continue sending that action potential and meeting that threshold. What we're going to kind of transition into is that cardiomyocyte action potential. And this graph, you notice, looks a little bit different. And so kind of right off the bat, just orienting you a little bit with this graph, you have the resting membrane potential of a cardiomyocyte is at more than -90 millivolts range. That has to do with the potassium leaky channels, which keep that potassium movement at about equilibrium, and that happens at about -90 millivolts when we put it in perspective of membrane potential. And so I mentioned the potassium leaky channels, so that's one significant kind of potassium channel that we have. We also have transient, and then we have our ordinary voltage-gated potassium channels, and we'll talk about what each of those do kind of moving forward here in a little bit. But you'll notice a lot of the process is similar. You still have depolarization from sodium moving into the cell quickly. You have repolarization from potassium moving out of the cell quickly. But what is special about cardiomyocytes is they have this plateau phase, and we'll spend a little time talking about that as we continue through this. So as I said, when a cardiomyocyte is just at rest, it's at about that -90 millivolt potential. And one of the other special things about the cardiomyocytes is instead of having that neural stimulation like you need in that skeletal muscle tissue or even in nerve impulses through the nervous system, your heart has what are called pacemaker cells. And these pacemaker cells are what are actually going to initiate that action potential and ultimately the contraction of those cells. So we have the potassium channels that are leaky, that are on when we are down at that -90 millivolts. And then a stimulation from the pacemaker cells is what is going to ultimately take us and lead us into that depolarization phase. So during depolarization, much like you have learned previously when you have looked at action potential, sodium voltage-gated channels open. This allows sodium to rush into the cell. Sodium is a positive ion, so as that positive ion goes into the cell, that is going to increase your membrane potential and essentially reverse it so that it has a positive membrane potential, and that sodium is going to continue to rush in until you hit about that +20 to 30 millivolt threshold again. similar. It just depends on which myocardial cell this is happening in. But at the peak, then, just like in our previous action potentials, our sodium gates are inactivated. But it's important to remember that they are not totally closed, they are just inactivated at this point. And so we still have our peak like you see on the graph, and now our cell starts to go into that kind of special phase. So when our cell hits that peak potential, so that very topmost point on the graph with where depolarization ends, that peak potential is going to trigger another class of potassium channels to open. So our leaky channels are open when it's at rest. Then depolarization happens. When we hit that peak, it opens some potassium channels that are called transient channels. And so that is going to allow a very quick but limited amount of potassium to get through those channels and kind of start the repolarization process, but not completely. Then, as we continue across our timeline here, we see that you have calcium now coming into play. Okay, so this plateau, before I go any further, this plateau phase that happens in these cardiac cells, are vital. This plateau phase has to happen in order for us to have that good quality contraction that is going to actually allow blood to be ejected from the heart and pumped through our body. So the plateau is unique to those cardiac cells because they're the only cells that have to do this and have to have that sustained action potential, that sustained phase of action potential, so that it can be a little bit longer of an action potential and allow for sustained contraction for the blood to be pumped without too much fatigue. So, all that being said, so we have had our little initial burst of potassium out. Now we have some channels for calcium that are going to allow calcium to move into the cell. And so as that calcium moves into the cell, our permeability for potassium to leave the cell has decreased because those transient gates are closed. We have calcium channels that have opened and are allowing calcium to flow into the cell at a fairly slow rate. And these two basically just counteract each other in terms of voltage. And so these calcium channels and the reduction in potassium are what is going to let us maintain this nice plateau of potential, so that we have our about 200 millisecond time frame where we are able to ensure that quality blood ejection from the heart before another contraction happens. One thing that I think is important to remember is we want this to be in that approximate time frame. So just like when we talk about homeostasis and any other aspect of our body, we want to make sure that our plateau phase is sustained so that it's as long as it's supposed to be, but we also don't want it to be too long. Studies have looked at diseased myocardium and issues that arise from that. Obviously, heart disease is a big problem in our country, but one of the things that has been looked at is what is causing, like when we see diseased myocardium, what's causing it. And on a molecular level here, we can look at, and many studies have been done surrounding the potassium and calcium influx and efflux as we are looking at this plateau phase. And what they have found is that while the calcium tends to be kind of the focus of many treatments, of many diagnoses. We also are noticing that this potassium and having this normal output of potassium is vital. And in diseased myocytes, sometimes we have the calcium issue, but alongside of that, we also have a problem with our potassium output. And so if we can recognize this, if we can look more closely into this, then we could see some different therapeutic treatments that could restore that normal potassium output and essentially restore function to those diseased myocardial cells without either without the need for treatments for the calcium or in combination with therapies that are treating that calcium deficiency as well. So ultimately, when we get through that plateau, we now are kind of moving back to what we've known before, in that in our repolarization phase of this action potential, we have calcium channels are now inactivated, so we're no longer allowing those calcium ions to move into the cell. Potassium is able to now rush out of the cell through those ordinary voltage-gated channels that we've seen before, and the cell is allowed to return to that resting membrane potential, and the potassium leaky channels open again, just as they did before the action potential began. This gives you kind of that reference for, while there are similarities between a cardiomyocyte and your typical skeletal cell that you've learned, skeletal muscle cell that you've learned about previously, there are some key differences, and you want to make sure you take note of those key differences and why they are so important to cardiac muscle function. Thank you so much for listening, and I hope you enjoyed it.